The popular understanding of the nervous system relies on an intuition of permanence. We imagine that the brain's connections are laid down in development, refined in youth, and remain structurally fixed thereafter. In reality, a synapse persists through a continuous maintenance subsidy. Its receptor complement, its cytoskeletal scaffold, and the dendritic spine that houses it are all in constant turnover, requiring ongoing support to remain standing. In the adult forebrain, the molecule that carries the largest share of that payment is Brain-Derived Neurotrophic Factor, or BDNF. When that subsidy is withdrawn, the synapse does not fail catastrophically. It fades, first in its capacity to strengthen, then in the stability of its spine, and finally in its very existence. This quiet, graded fading of synapses is the strongest structural correlate of cognitive decline in Alzheimer's disease. Synapse loss is a trophic bankruptcy, an account that simply stopped being paid. Among the molecular findings in Alzheimer's disease, one observation is entirely uncontested across 3 decades of study. BDNF messenger RNA and protein are significantly reduced in the Alzheimer's hippocampus and cortex. Crucially, this reduction is present in the preclinical and prodromal stages of the disease. The deficit is established while there is still a vast amount of living brain tissue left to protect. But around this solid observation gathers the governing difficulty of the entire scientific field, the threat of reverse causation. This UI lays out the 2 competing interpretations. The first argues that withdrawing trophic support actively kills the synapse. The second, the Tombstone hypothesis, suggests that dead neurons simply stop producing BDNF, making the loss a mathematical consequence of cell death rather than its cause. If the Tombstone reading is correct, low BDNF is entirely off the causal path. It is a passive marker recording the destruction, making BDNF an irrelevant target for any therapeutic intervention. To prove that the loss of BDNF actively drives cognitive destruction, we must defeat the Tombstone null hypothesis. We must grade every biological connection in a strict validity ledger, separating consequences of the disease from the actual causes. We start with the baseline physiology of the healthy brain. BDNF is an activity-dependent neurotrophin. A neuron must depolarize and fire to produce and secrete it. Neuronal activity drives CREB. CREB produces BDNF, binding the TRKB receptor, initiating a cascade that loops back to drive more CREB. This trophic support sustains the machinery for activity, the embodiment of Hebbian maintenance. But this positive feedback loop is vulnerable, perfectly capable of amplifying downward. The brain's central mechanism for structural plasticity and learning presents a wide-open biological target for Alzheimer's pathology to exploit. To pass the validity ledger, we have to look at how Alzheimer's pathology interacts with living neurons upstream of their death. Returning to the circuit, we introduce amyloid beta oligomers. These clusters interfere directly with CREB phosphorylation, suppressing the specific promoters that activity would otherwise turn up. Amyloid drives this trophic decline in living cells, a primary lesion, completely separate from the arithmetic of cell loss. The disease also attacks the composition of the neurotrophin itself. BDNF is synthesized as a precursor, pro-BDNF, which is cleaved to yield the mature protein. These 2 forms dictate entirely opposing commands. Mature BDNF binds the TRKB receptor to signal survival and growth. The uncleaved precursor, pro-BDNF, binds a different receptor, P75NTR, to drive synaptic depression and apoptosis. This scale maps the critical ratio between the 2 forms. In the Alzheimer brain, the cleavage process is impaired, forcing the balance heavily toward the uncleaved precursor and its destructive P75NTR receptor. The disease reduces the available subsidy and actively corrupts the remaining currency into a neurotoxin. Even when mature BDNF is successfully secreted and bound at the synapse, it cannot deliver its survival signal without being physically transported back down the axon to the cell body. This axon cross-section displays parallel microtubule tracks. Tau pathology destroys this cytoskeletal scaffolding, severing the retrograde transport of the neurotrophin payload. Caught in a pincer movement, amyloid throttles BDNF synthesis, while tau severs its delivery. The soma is starved of its survival signal. The positive feedback loop of healthy plasticity is completely inverted. Less trophic support weakens the synapse, leading to less neuronal activity, which in turn drives even less BDNF transcription. The descent becomes self-sustaining. The trophic deficit outruns and outlasts the amyloid burden that initiated it. The biological mechanisms are coherent, but correlations in an autopsy brain cannot decisively resolve the reverse causation problem. The final test for the validity ledger is interventional. If BDNF is truly a passive tombstone, supplying artificial BDNF to a brain that is losing it for other reasons should accomplish nothing. Researchers delivered AAV-BDNF gene therapy and small molecule TKB agonists into the brains of aged primates and amyloid-bearing rodents. The introduction of the trophic factor rescued learning, improved memory, and stabilized synaptic markers across the models. Looking at the final row of the validity ledger, we find the critical caveat. This functional rescue occurred entirely downstream of an unchanged amyloid plaque burden. Supplying the missing BDNF protected the synapse despite the continued presence of the primary pathology. By improving function while the upstream disease remains, the interventional data isolates BDNF loss as an active driver of cognitive destruction. The Tombstone hypothesis is falsified. The activity-dependent nature of this biology translates directly into clinical resilience through the concept of trophic reserve. Interventions like environmental enrichment and aerobic physical exercise reliably elevate BDNF expression. Generating higher baseline levels of trophic support banks molecular capital against future pathology, subsidizing the synapse so it can withstand a heavier toxic load. This mechanism carries a specific human genetic handle. A common germline variant in the BDNF gene, known as Val66Met, impairs a neuron's ability to secrete BDNF specifically in response to activity. This chart plotting cognitive decline over time demonstrates the gene-by-pathology interaction. In cognitively normal individuals, carrying the variant barely registers. But once the amyloid-positive threshold is crossed, the variant violently accelerates the rate of decline. The genetic variant independently lowers activity-dependent BDNF secretion. When amyloid arrives and begins suppressing BDNF transcription, both the germline and the pathology strike the identical trophic vulnerability at the same time. This structural interaction explains why two brains with identical plaque burdens can host minds that are decades apart in functional capability. Between the upstream lesions of Alzheimer's and the final disappearance of the mind, there is a trophic economy. The disease dismantles the brain, in large part, by slowly withdrawing a vital synaptic subsidy. This imposes a strict temporal constraint on any therapeutic attempt to replace it. Restoring a trophic factor is a maintenance intervention. It is not a resurrection. This timeline of Alzheimer's progression maps the therapeutic window for BDNF gene therapies and TKB agonists. The bracket opens early in the preclinical and prodromal phases. The therapy can preserve synapses that are stressed but still physically present. It cannot rebuild circuits from neurons that are already gone. Earlier trials across neurodegeneration failed because they expected a maintenance molecule to reverse established dementia. They tried to pay a debt on a closed account. Synapse loss in Alzheimer's is a trophic bankruptcy. It is a biological debt that can finally be paid, provided we intervene while the synapse still stands to receive it.